Welcome to the Vet NeuroJar podcast. Today, we are diving into a fascinating research article titled Adeno-Associated Virus 8 and 9 Myofiber type/size, Tropism, Profiling Reveals Therapeutic Effect of Microdystrophin in Canines. This study, conducted by Matthew J. Burke and colleagues at the University of Missouri and Indiana University, explores gene therapy for Duchenne Muscular Dystrophy, or DMD, using adeno-associated viruses, or AAVs, in dogs. Background and significance. Duchenne muscular dystrophy is a devastating inherited neuromuscular disease. It is characterized by progressive muscle weakness and atrophy. The disease results from mutations in the dystrophin gene, which is essential for muscle fiber integrity. Current treatments offer limited relief, making gene therapy a promising avenue for therapeutic intervention. Adeno-associated viruses, specifically serotypes 8 and 9, are being evaluated in clinical trials for treating neuromuscular diseases, such as DMD. These viruses are used as vectors to deliver therapeutic genes into muscle cells. However, muscles are composed of different types and sizes of myofibers, and little is known about how these viruses interact with these varying muscle fibers, particularly in large mammals. The success of adeno-associated virus gene therapy relies heavily on the tissue and cell type specificity of the viral capsids. This study aims to understand the fiber type and size preferences of adeno-associated virus 8 and 9 in dogs, providing crucial insights for optimizing gene therapy strategies for DMD. Methods and experimental design. The researchers conducted a comprehensive study involving 17 dogs. These dogs received systemic gene transfer with adeno-associated virus 8 or 9 vectors. The average dose was 1.94 by 10 to the 14th vector genomes per kilogram. The dogs were injected at approximately 2.86 months of age and harvested at about 20.79 months. 4 adeno-associated virus 8, 2 DMD-affected dogs, and 3 carrier dogs received an alkaline phosphatase reporter vector. 5 DMD-affected dogs received a 4-repeat microdystrophin vector. 4 adeno-associated virus 9, 1 normal, and 1 DMD-affected dog received the alkaline phosphatase vector. Additionally, 5 DMD-affected dogs received a 5-repeat microdystrophin vector. The scientists evaluated transgene expression in 3 muscles that exhibited mosaic transgene expression. These muscles included the biceps femoris, teres major, and latissimus dorsi. Mosaic transgene expression refers to the pattern where some muscle fibers express the transgene while others do not. To correlate transgene expression with muscle fiber type and size, the team used immunofluorescence and histochemical staining. Muscle samples were frozen and cryosections were stained for alkaline phosphatase activity. They were also immunostained for microdystrophin, laminin, and myofiber types. Antibodies were utilized for immunostaining, and the slides were visualized using a fluorescence microscope. Myofiber type and size were quantified using specialized software, with manual validation of the results. The analysis included quantifying the overall fiber type composition in each muscle sample. This was done by calculating the percentage of each fiber type relative to the total number of fibers. The fiber type composition of transgene-positive myofibers was also quantified, along with the percentage of transgene-positive myofibers in each fiber type. Key findings. The study revealed several significant findings. Transgene expression was detected in 30 to 45% of myofibers. In the dogs injected with the alkaline phosphatase reporter vector, neither adeno-associated virus 8 nor 9 showed a statistically significant fiber type preference. In the microdystrophin-treated DMD-affected dogs, slow and fast myofibers were equally transduced. Notably, myofibers expressing microdystrophin were significantly larger than those not expressing microdystrophin, irrespective of the adeno-associated virus serotype. Specifically, in adeno-associated virus 8 microdystrophin vector-injected dogs, the mini-ferret diameter, a measure of fiber size, was 15%, 16%, and 23% larger in microdystrophin-positive slow, fast, and hybrid fibers, respectively. The cross-sectional area, another measure of fiber size, was 30%, 34%, and 46% larger in microdystrophin-positive slow, fast, and hybrid fibers, respectively. In adeno-associated virus 9 microdystrophin vector-injected dogs, the The mini-ferret diameter was 12%, 13%, and 25% larger in microdystrophin-positive slow, fast, and hybrid fibers, respectively. The cross-sectional area was 25%, 28%, and 59% larger in microdystrophin-positive slow, fast, and hybrid fibers, respectively. Detailed analysis of fiber types and transgene expression. The research delved deeper into the characteristics of different myofiber types and their response to adeno-associated virus-mediated gene transfer. Skeletal muscle consists of myofibers with distinctive phenotypes, including type 1, type 2, and hybrid myofibers. Type 1 fibers express slow myosin heavy chain, while type 2 fibers express fast myosin heavy chain. Hybrid fibers express both slow and fast myosin heavy chains. Type 2 fibers can be further divided into types IIa, IIb, and I9. Type 1 fibers are slow oxidative fibers, generating smaller force but resisting fatigue. Type IIa fibers are fast oxidative/glycolytic fibers, generating moderately high force at a relatively fast rate and moderately resistant to fatigue. Type IIb fibers are fast glycolytic fibers, generating very high force at a very fast rate, but fatigue quickly. Type I9 fibers are also fast glycolytic fibers, exhibiting a contractile profile intermediate between type IIa and IIb fibers. In the Adeno-Associated virus alkaline phosphatase vector injected dogs, the researchers detected type 1, type IIa/I9, and hybrid fibers, but not type IIb fibers. This aligns with the understanding that dog muscles, similar to human muscles, lack type IIb fibers, unlike mouse muscles. The study found that Adeno-Associated virus 8 and 9 transduce slow and fast myofibers at similar efficiency. This suggests that these viral serotypes do not have a strong preference for a particular fiber type in canine muscles. Impact of microdystrophin therapy on myofiber size. A crucial aspect of the study was evaluating the impact of microdystrophin gene therapy on myofiber size in DMD-affected dogs. The absence of dystrophin leads to muscle atrophy, and strategies to increase myofiber size are actively being pursued. The researchers compared myofiber size between normal and affected dogs that did not receive adeno-associated virus injection. They also compared microdystrophin positive and negative fibers in adeno-associated virus microdystrophin-treated affected dogs. As expected, the fiber size of affected dogs was significantly smaller than that of normal dogs. However, in adeno-associated virus microdystrophin-treated affected dogs, microdystrophin-expressing myofibers were significantly larger than those without microdystrophin expression. This indicates that microdystrophin therapy can prevent myofiber atrophy in dystrophic dogs. Discussion and implications. The study provides valuable insights into the fiber type and size tropism of adeno-associated virus 8 and 9 in dog muscles. The finding that these serotypes transduce fast and slow fibers at similar efficiency is crucial for designing effective gene therapy strategies for DMD. The observation that microdystrophin therapy prevents myofiber atrophy further supports the therapeutic potential of this approach. The researchers acknowledged several limitations in their study. These include variations in the age of dogs at injection and harvest, the use of different promoters for alkaline phosphatase and microdystrophin expression, and a small sample size, particularly for the adeno-associated virus 9 alkaline phosphatase group.